Okay, our final presenter before lunch is Dr. Clive Svendsen, Director of Cedars-Sinai Regenerative Medicine Institute. Clive?
Okay, great. Thanks very much for hanging in there. The last one, I'm between you and lunch, so I'll be very concise. I don't have a company, a startup company. This is my partner, Cedars-Sinai Hospital. Thought I'd tell you a little bit about Cedars-Sinai. Many of you know it as a hospital to the stars in Beverly Hills. But recently, we've put a lot of energy into the research mission here. This is the research tower at Cedars. And we're ranked highly amongst hospitals nationwide. We have over 800 externally funded projects, $100 million of research funding, and 800 employees involved with research. So we're really building the research mission at Cedars-Sinai. And over the last even 6 years, the research has been growing very, very rapidly within this medical center. And I think I'll come back to this. Medical centers are actually a good target, a good site for doing these phase 1 type of clinical trials under the umbrella of an institution like Cedars. Regenerative Medicine Institute was opened back in 2010. Involves 13 principal investigators, which work together for bringing therapies to the clinic as one of our goals. The brain, we have a brain program, a pancreas liver program, blood and eye program, and a skeletal program. And these work together with other institutes on campus, including the Heart Institute with Eduardo Marban, to synergize the energies and try and get stem cell treatments through to patients. The core facility, an IPS core facility, makes IPS lines. That's one of our main focuses. And our mission is to provide cells to clinicians and researchers. And I think, again, the advantages are of being an academic center. We can do these trials and go all the way from bench to bedside within the institute. We actually have a graduate program that's just been accredited now in Cedars-Sinai, and outstanding core facilities. And the research we're doing is highly translational in areas, as I'll discuss in a moment, ALS, but also bone, osteoporosis, macular degeneration, and corneal diseases. So some of the highlights of the Institute recently, and I think this is just the excitement of IPS cells, is you can make any tissue of the body. And in collaboration with Steph Targan, we start making gut organoids, and we're looking at using these models for IBD that Steph works on. We just published a model of Huntington's disease, and this is the other power of IPS, as you just heard, is disease modeling. And we now have a pretty good functional model of Huntington's disease in the dish. We just were awarded through CERM, first collaborative grant, I think, with CERM and NIH, which brought together these two institutions to make another 15 IPS lines from Huntington's disease. And so this is nice to expand that work. And we also have a couple of new grants to look at Alzheimer's and corneal diseases using IPS cells. So those are kind of an overview of the Institute. What I'm going to talk about now is ALS and the latest grant that we got. Again, this is the cadre of CERM grants. We were awarded a grant from CERM very recently to push cells forward for ALS. So which cell types are we using? Well, you've heard a lot about mesenchymal stem cells from the bone marrow, and of course these are very useful in general for treating disease. I think we're all agreed that in very few cases in studies that have been done, do these cells really replace lost cells in disease. Most of the time they probably release growth factors and modulate the immune system. So this isn't a cell replacement therapy in general. It's more a modulation therapy. To actually replace cells, as you've heard, probably going to have to use a different source, and I think it's going to either probably be embryonic stem cells or IPS cells. As you heard, embryonic stem cells and IPS cells are available for the brain. We want to take them into a progenitor state and then make them into the 3 cell types of the brain, which we're interested in. For my money, replacing whole neurons in the brain is going to be incredibly difficult. And so neuronal replacement in Parkinson's disease has turned out to be more complex than we thought. If you get the wrong neuron in the wrong place, you actually get side effects and dyskinesias. So the focus really in the field, as you heard today, is remyelination from oligodendrocytes, which is something you can do with the intact circuitry. And what we've been focusing on, a number of other groups, is to replace the astrocytes to support cells in the brain. So I think in the future, we may be able to reconnect pathways, but right now, the focus is more on astrocytes. And of course, these cells have a teratoma risk because of with great power comes great responsibility. So I think the other option is fetal tissue, and that's the cell source that we've been using recently. And I'll explain briefly the type of cells that we've been using and 2 other companies that you've heard from. And these cells are got from human postmortem fetal tissue at about 90 to 100 days. We expand them up in EGF and FGF. And then at later passages, they actually make astrocytes, which is the cell type we want. And you heard from two other companies during this meeting, Stem Cells Inc. and Q Therapeutics. And both these companies use a similar product, actually. Stem Cells Inc. sort their cells in CD133 sorting and use them before passage 10. And Q Therapeutics design a glial progenitor, a different type of cell that only makes astrocytes and oligodendrocytes. But again, these are early passage cells. And another company called NeuralStem, again, early passage. The unique aspect of our cell type is at late passage. These cells can be expanded to huge numbers, and we can make working lots of a product to treat hundreds or thousands of patients from the same starting fetal material. The other thing to bring in is GDNF, and I've had a long-standing interest in this compound, this drug or growth factor. In Parkinson's disease, we showed a number of years ago we can infuse this into patients safely with Parkinson's disease, and it's seen to have pretty astounding effects on metabolism in the brain, dopamine in the brain. It's a growth factor that's pleiotropic. It acts on lots of different nervous systems and nervous system neurons. And so from there, Amgen actually took this on, and it's a long story, but our interest was actually taking those cells I just described, using viruses to infect them, and making stem cells that produce GDNF. So now we have a combined product, which is the neural progenitor, neural stem cell, engineered to make GDNF. And this is our combined product that we're interested in taking forward into the clinic. The disease is ALS, sometimes known as Lou Gehrig's disease because of the baseball player in this country that got it. There's no cure for ALS. So you heard from a number of disorders today. There's no treatment. And the idea is that the stem cells would be made into support cells, injected into the spinal cord, releasing GDNF. And this should slow down the degeneration of the disease based on all our preclinical models. The CIRM award that we got was based on this hypothesis, really using neural stem cells that I described to replace the sick astrocytes around the dying motor neurons to limit and slow down the progression of motor neuron loss in the disease. The work plan is basically, I'll go through briefly, but it's GMP-grade cells that we will be working in. The team is Larry Coulter from City of Hope, and then we're going to infect them with the clinical-grade lentivirus that we had made by the National Gene Vector Labs when they were still alive, and these viruses are already made, and we moved to IND filing within 2 years, and we have a trial design, and it's a collaborative grant between 3 sites here at Cedars-Sinai, and then Emory and California Pacific Medical Center with the following collaborators. We should be to IND at around a year and a half, it's 2014, and then complete the clinical trial in 2016. The master cell bank has already been produced actually at the University of Wisconsin when I was there with Derek High, and we're going to take that bank and make 3 lots, a research process comparable lot and a GMP-grade clinical lot. This is the process. We just expand up, as you all know, through this process. In the middle, we do an infection with lentivirus, clinical-grade lentivirus. The neat thing is when you use lentivirus, that these cells then, when they divide, carry the lentivirus and will make your drug for you in the dish. So we can keep the cells dividing, and they continue to reach GDNF at stable levels. And then we go through the normal series of preclinical work that we need to do, and we've established these milestones with CIRM. And we're about to move forward. We're going to have the maximal feasible dose that we do in the SOD1 model, which is a model of ALS. We're going to use SPIO labeling and some other techniques based on some interactions I've had here at the meeting with a number of companies. There may be a better way to do this, particularly Bell Companies is interested in helping us with this. Then we'll perform safety tox studies at Explorer Biolabs, which is a CRO, tumorigenicity studies again there, and finally a dose escalation study in pigs, mini pigs. And I love pigs, by the way. And it's very sad that we have to use any animals in research. And then we'll do the dose escalation in pigs, which have a spinal cord of similar size to the human cord. We've done a lot of work on this previously in collaboration with Emory and Nick Boulis and others. So we are very confident we can get these cells in. We have a clinically a device here at the bottom that's been used clinically already in patients at Emory to deliver cells to the spinal cord. And we have strong preclinical data showing that the GDNF secreting cells actually save motor neurons. So here's a transplant in the spinal cord, and here you can see the side that had the cells, and you can appreciate they still have lots of motor neurons, whereas the side that didn't get the cell had none. And then finally, the trial design. It's kind of a unique design. It's an 18-patient trial, but the neurosurgeon, when they go in and do the laminectomy, are going to look at the blood vessel structure on the cord and decide to do either right or left cord. These cells don't migrate across the midline. So these will be unilateral transplants. And in patients, if they get ALS in one leg, it then spreads to the next one pretty soon, and then it goes to your arms, and eventually you're paralyzed, and final thing to go usually is breathing. So if one leg is affected, the second leg will proceed at about the same rate. So because the cells don't cross the midline, we can implant them in one side of the spinal cord and assess very carefully degeneration or lack of function in that leg over time, and then the other leg is used as control because that didn't have a transplant. So that way you get much more power of the analysis, and we can design it as a 2A to get efficacy data from this very small group of patients. And that's the unique thing about giving stem cells regionally to one part of the CNS, that you can get regionally specific data, because the GDNF is only made where you put the cells. So this, we feel, is a very powerful design. Interesting, the neurosurgeon will decide, but he's not going to tell the neurologist. So the neurologist will be blinded as to what side the patient got, and the patient will be blinded as well. So this is also a blinded trial. If there's any effect from a data monitoring safety committee, at 6 months we can, in a year, we can perform a crossover whereby the other side can get transplanted. So that's the design of the trials, the first in man for this type of approach. And finally, we're partnering with a company called Unicure, who have one of the first gene therapy patents in Europe, who hold the licenses for GDNF that Amgen sold them a few years ago. And they're willing to take this forward with us should we be successful in this trial. And the final slide, you know, we've worked out this for ALS, but as you heard from Neuralstem in the first presentation of this session, these cells are pretty pleiotropic. They can go into different disease models. And GDNF is also pleiotropic. It acts on different systems. So we have an active program in Huntington's disease based on publications there that they protect neurons. Macular degeneration with Xiaomei Wang, who's behind a lot of the preclinical data for the trials you heard in macular degeneration. We recruited her to Cedars-Sinai, and so she and I have collaborated before to show these cells can work in macular degeneration. In stroke with Gary Steinberg, good friend of mine. We just had a paper in Brain showing these cells can reduce damage in stroke and increase plasticity. And finally, Parkinson's, where I started getting interested in this field. We have a lot of history in Parkinson's, and we're about to reactivate Parkinson's module of putting the cells in for Parkinson's disease. So this is a new building going up at Cedars-Sinai. It's a $600 million glass tower. That's the Advanced Health Sciences Pavilion. It just shows you how Cedars is totally invested in making these kind of therapies work for the heart with the Heart Institute and now the Regenerative Medicine Institute. And so it's nice to have them partnering with us through this process. And I think you hear from Capricorn later on, who are also in Cedars and working very closely with us to try and get these therapies into the clinic. So exciting. Disease team proposal has been fantastic for us. I think medical centers such as Cedars are a very good place for you guys to come and invest in and work with, because I think ultimately I'd rather this went straight to being reimbursed rather than going through the company route. I know nobody makes any money that way, but I think this is a model that perhaps will work for these incredibly complex strategies of neurosurgeons, neurologists, and going all the way through to patients. And so that might be a way that we can get cells through to the clinic quickly. And I hope that we're on the road to a therapy for ALS and other disorders. So thanks very much. And lunchtime.